Hello everyone, welcome back. In the last two episodes, we talked about the resting membrane potential. Now, it's time to wake them up and throw them into some action. In these two episodes, we are going to talk about the action potential. We're going to learn about the whole process of the action potential. Let's take a look at the overview. First, we are going to start again with proteins, especially voltage-gated ion channels. And then in this half episode, we are going to talk about all the stages of an action potential. In the next episode, we will see the effect of drugs on action potentials. We will talk about the conduction and transmission of action potentials, then how to measure the membrane potential, and we will introduce a model, the Hodgkin-Huxley model, that uses mathematics to describe the behavior of neurons. Let's start with voltage-gated ion channels. As you may remember, this is also a type of facilitated diffusion. However, these ion channels are gated by the voltage of the membrane. This is a voltage-gated sodium channel. As an example, when the membrane potential is at minus 65 microvolts, the channel is closed. However, when the membrane potential changes to minus 50 microvolts, then some parts of the protein gets pushed up and the channel opens. We also have voltage-gated potassium channels, voltage-gated calcium channels, and they're all voltage-gated, of course. How? How does voltage open the ion channel? Let's take a look in detail. First of all, like all other ion channels, voltage-gated ion channels have pore loops to ensure selectivity. As you may recall, selectivity means that the ion channel only has high permeability for one ion and do not let other ions to pass through. Moreover, voltage-gated ion channels have an interesting structure. It is a voltage-sensitive helix. As we can see in this figure, the full-length voltage-gated ion channel is made of 4 subunits. They are placed side by side here, but in reality, they form a tetramer, like this. Each individual subunit is made of many helices with a pore loop. The fourth helix, marked in purple here, is especially important. It is the voltage-sensitive alpha helix. How do we accomplish that? Well, this voltage-sensitive helix has many charged amino acids on it, especially amino acids with positive charges. So when the membrane potential is negative enough, it is pulled down towards the intracellular side. However, when the membrane potential increases, the inner side of the membrane becomes more positive, and this pushes this helix up towards the out of the cell. The movement of this helix propels the whole protein to change in conformation, thus opening up the channel and allowing ions to flow through. Moreover, the voltage-gated sodium channel has one other interesting feature that other voltage-gated channels lack. That is the ball and chain structure. Thanks to the ball and chain structure, the voltage-gated sodium channel has 3 different states. The first state is the closed state, where the channel itself is closed. The second state is the open state, where the channel is open and the ball is freely dangling outside. The third state is the inactivated state, where the ball is plugged in to the open ion channel, thus blocking the flow of ions. And lastly, the protein returns to the closed state, where the ball flows down and the ion channel again closes. We will represent this by this circle diagram. C represents closed, O represents open, and I represents inactivated. This little graph will be very useful in the next section about the different stages of the action potential. A special type of voltage-gated potassium channel is also very interesting. It is called a delayed rectifier. Never mind what rectifier means for now, but focus on the word delayed. It is called delayed because its reaction is very slow. So, after the voltage change, after the voltage reaches the threshold, that is to say, when the channel can be opened by the voltage, it does not open immediately, but rather it waits 1 millisecond before it opens. That is why it is called a delayed rectifier. Question time. Which is not one of the three states of the voltage-gated sodium channel? A. Channel open with ball pulled out. B. Channel open with ball plugged in. C. Channel closed with ball pulled out. And D. Channel closed with ball plugged in. That's right, D. Channel closed with ball plugged in. This is impossible. A is the open state, B is the inactivated state, and C is the closed state. Now it's time to get to the action potential. You will encounter many new terms in this slide, but don't worry, either they're old friends or they're just really easy to understand. So first, this is the graph of an action potential. The horizontal axis is time, and the vertical axis is the membrane potential. Starting at -70 microvolts, we have the first segment, resting state. It corresponds to number 5 in the graph. Then, after a stimulus is received, we get to the depolarization, or rising phase. This corresponds to number 2 in the graph, where the membrane potential rises to positive. Thirdly, we have repolarization, or falling phase, where the membrane potential falls again to -70 microvolts. After that, we get hyperpolarization, where the membrane potential dives down below -70 microvolts. And resting state again. We also have another set of terms for these. First, after the resting state, when the stimuli is received, we need to get to the threshold. The threshold is a specific value, and we need to get to it to initiate an action potential, ergo the name initiation. We also have the overshoot. This refers to one part of the rising phase and a part of the falling phase in which the membrane potential is greater than 0. After the repolarization, we have the refractory period. Let's get started with the resting state. In the resting state, the neuron is sound asleep. It is not responding to any stimuli, and the membrane potential is maintained at -70 microvolts, the resting membrane potential that we calculated in the last episode. In this state, the voltage-gated sodium channel is currently closed. Now, initiation. In this phase, the neuron receives a stimulus and it begins to depolarize. That is to say, the membrane potential tries to become less negative. We need to reach a voltage that is required to open the voltage-gated sodium channel, and that is usually considered to be -55 microvolts. If we fail to reach the threshold, as in the figure, we have failed initiations. In this case, the closed-state voltage-gated sodium channel is unable to fully turn into the open state. However, if we do reach the threshold, then the closed state will successfully transform into the open state. Where does initiation happen? In the spike initiation zone. The spike is a term to describe the action potential because, as we can see, the action potential has a very sharp spike-like shape. Thus, we call one action potential a spike. This zone can be located at different locations in different neurons. In most neurons, it is the axon hillock. As you may recall, this is the part where the axon leaves the soma. The dendrites collect signals and conduct spatial and temporal summations. That is to say, the signals are added together by space and with time. If the total signal can reach the threshold, then the spike is initiated at the spike initiation zone, the axon hillock, and the signal is sent down the axis all the way to the terminal bouton. In sensory neurons, however, the action potentials can be initiated at sensory nerve endings. These are endings that are very rich in certain types of gated ion channels or other proteins. For example, pressure-gated ion channels are located at the end of some somatosensory neurons, neurons that sense touch. If it is touched, then the sensory nerve ending will initiate the spike, and that passes down all the way to the terminal bouton of the axon. After initiation, we have the depolarization, or by the shape of the curve, we also call it the rising phase. In this phase, the voltage-gated sodium channels stay open. There's actually a positive feedback in this process. As sodium ions flow into the cell, the membrane potential rises, and even more voltage-gated sodium channels open. The influx of the sodium ions is due to the concentration gradient. There is a higher concentration outside than inside, so sodium ions flow in. In the overshoot, the membrane potential becomes positive. That is to say, the membrane polarity is reversed. It turns from negative to positive. After about 1 millisecond after the initiation of the spike, the voltage-gated sodium channels begin to close. Well, it is not so accurate to say that they are closed, but rather we should say that they are inactivated. That is to say, the ball is now stuck into the open ion channel to block the flowing of the ions. After that comes the repolarization, or by the shape of the curve, we also call it the falling phase. The sodium ion channels still stay in the inactivated state, while the potassium channels, the delayed rectifier voltage-gated potassium channels, finally open after 1 millisecond. Wow, he's finally here. From this stage, we can finally see the meaning of rectifier. These channels rectify the membrane potential in that it pulls the membrane potential back close to the resting potential. There is an efflux of potassium ions. That is because the concentration of potassium ions is higher on the inside than the outside. Thus, the neuronal membrane begins to repolarize, and then it hyperpolarizes. That is because the influx of potassium ions fails to break immediately, but rather continues even after the neuronal membrane reaches minus 70 microvolts. Recall that the equilibrium potential for potassium ions is around minus 80 microvolts. That is why, at most, the membrane potential could reach minus 80 microvolts. After a while, the influx of potassium stops, as we can see in the local minimum of the graph, and excessive potassium ions begin to leak out, returning the neuron to resting state, finally. Now, what does the refractory period mean? The word refractory comes from refrain. The neuron refrains from another action potential. There are two types of refractory periods, absolute and relative. In the absolute refractory period, marked in red in this graph here, it is impossible to initiate another action potential. That is because sodium channels are inactivated and they cannot direct go to the open state. In the relative refractory period, however, it is only difficult but not impossible to initiate another action potential. And that is because the membrane is hyperpolarized and we need more stimuli to reach the threshold again. Finally, the inactivated sodium channels go themselves back to the closed state, and they are now ready to open up again. The resting membrane potential is returned. In this process, two key proteins have their functions. As I've said before, the potassium leak channels return the protein by leaking potassium out of the cell, and the sodium-potassium pump maintains the concentration gradient of both sodium and potassium. Now it's time for a review of the whole process of the action potential. Please try to put the following events in chronological order. Some of them are a little bit tricky and I have put them in bold font. Event 1. Sodium ions flow in even though electrostatic force tries to push it out. 2. Voltage-gated potassium channels close. 3. The polarity of the membrane is reversed. 4. The membrane potential reaches minus 55 microvolts. 5. Voltage-gated sodium channels open. 6. The membrane potential hits minus 70 microvolts for the first time after the action potential is initiated. And 7. Sodium channels become inactivated. Please take some time to pause and think about it. If you'd like to, you can take a paper and write it down. This is the answer. First, the membrane potential reaches -55 microvolts. It reaches the threshold and the action potential is initiated. Second, the voltage-gated sodium channels open up. Thirdly, the polarity of the membrane is reversed as the overshoot is reached. Fourth, sodium ions flow in even though electrostatic force tries to push it out. This is because the polarity of the membrane is reversed, and now the membrane potential inside the cell is positive, so the electrostatic force tries to push the sodium ions out. However, due to the concentration gradient, sodium ions are still flowing in. 5. Sodium channels are inactivated. 6. The membrane potential hits -70 microvolts for the first time after the action potential is initiated. As you may recall, this is between the repolarization phase and the hyperpolarization phase. And 7. The voltage-gated potassium channels close in the middle of the hyperpolarization phase. In this episode, we learned about voltage-gated ion channels and took a very detailed look at the stages of an action potential. In the next episode, we are going to talk about the effect of drugs on the action potential, how the action potential propagates after its initiation, how to measure the membrane potential, and give a brief introduction to the Hodgkin-Huxley mathematical model. That is all for this period. In the next period, we will talk about more about the action potential. See you again then.